Hi everyone, Professor Stefero here, and this screencast is going to discuss proteins. Proteins are very important in biological organisms. They perform many vital functions. They have several characteristics. Some proteins serve as transport proteins, and here they would carry other molecules. So hemoglobin is an example of a transport protein. It carries oxygen in the blood. Others are called regulatory proteins. These would regulate biological processes, so things like hormones and neurotransmitters are regulatory proteins. Contractile proteins are the muscle proteins that are found in the cells that are muscle cells, and they provide movement. Some provide structure, so proteins like keratin and collagen. Keratin we find in our hair and our fingernails, and collagen is found in the skin, and so it's to provide structure and support for those anatomical parts. Others are enzymatic proteins that are going to catalyze biological reactions, and an amylase is an example. Amylase is the enzyme that will help to begin to break down starch in your mouth, secreted by the salivary glands. Other proteins serve as antibodies that help defend against infection. And then finally, the last group would be storage proteins. And storage proteins are things like albumin in egg white, providing a ready source of amino acid for the developing egg. So proteins are a polymer made up of a sequence of individual amino acids that are bound to each other. And the sequence of amino acids determines the structure and the function of the protein. And there are 20 different amino acids, and generally all amino acids have 3 parts. The first part is called the amino group, and this is a functional group that contains the nitrogen atom. The second is the carboxylic acid group, and it contains a carbon double bonded to an oxygen along with the hydroxyl functional group. Then we have what's called the R group, or R side chain. This is the group that's going to be different for each of the 20 amino acids. And so we use R here to represent this generic side chain that's going to actually represent different structures depending on the amino acid we're talking about. So let me show you an example of that. So here we see some amino acids: glycine, aspartic acid, lysine, and cytosine. And if we look at the beige boxes here, they will all have the same structure, which is the nitrogen group and the carboxylic acid group. But if we look at the green is what's representing our R group, or our R side chain, and see how each one is different depending on which amino acid we're talking about. And that is what we mean by a difference in the amino acids. So it's the R group that's going to become really important to amino acids because it's going to determine the chemical characteristics of any amino acids. So some amino acids, by virtue of that R side chain, will be what we call hydrophilic. And hydrophilic refers to something that's water-loving. So a water-loving amino acid can dissolve easily in a water-based or aqueous solution. The other type would be hydrophobic, and hydrophobic is water-hating or water-fearing. And these sorts of proteins will not dissolve easily in an aqueous solution. But any protein is going to be made up of both hydrophilic and hydrophobic amino acids. And that's important because ultimately that's going to help build the three-dimensional functional structure of our amino acids. Here we can see the amino acids divided up even more. Some of them, because of the R group, are nonpolar. If you look at the blue now, in each case the blue shows those nonpolar branches that coming off of the main part of our amino acid, right? Lots of carbons, lots of hydrogens as a part of those groups. Then down here, the polar amino acids are going to have hydroxyl groups and other functional groups that make them more hydrophilic. Then there are going to be some that are going to be electrically charged, and those are here. We'll see the acidic type, so aspartic acid and glutamic acid. They have the carboxylic acid functional group here. We're showing the ionized version of this at the moment, so that's why the hydrogen is missing off of this particular molecule. And then some are basic, and the functional group that gives them that basic structure is that amino group. So again, we see different amino acids with different characteristics based upon that R functional group. When amino acids come together to begin to build a protein, they go through the dehydration synthesis reaction and create what's called a peptide bond. The peptide bond occurs between the carboxylic acid of one amino acid and the amine group of the other, and the peptide bond is then found between them. And this would be the special kind of bond that's found in a protein. So here we see the balls here with the 3-letter abbreviations. Each ball is an amino acid. They are next to each other, and so that represents the peptide bond of a particular protein. So this protein is insulin, and it's showing the unique structure associated with it. But it's going to take hundreds or 1,000 amino acids to build a 3-dimensionally functional protein. And it all starts with the amino acid sequence. So let's take a protein called transthyretin. Transthyretin is a normal blood protein. Its function is to carry thyroid hormone, so it would be an example of a transport protein. And if we start by looking at the layers of structure of this protein, we'll get a feel of how all proteins are made. So let's start with what's called the primary structure.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function.
So the primary structure is the amino acid sequence, and amino acids are linked to each other through peptide bonds. And here's why this is significant. Here's the amino acid sequence of a piece of the hemoglobin protein. And here we see the abbreviations for individual amino acids. But in sickle cell anemia, one amino acid is different in the structure of hemoglobin, and that leads to a non-functional hemoglobin and results in sickle cell blood cells. And sickle cell anemia and sickle cell blood cells are quite important because the cell is not the right shape, and it starts to clog up tiny blood vessels throughout the body and leads to organ damage. So just one change in one amino acid can affect the function of a protein. Let's talk about the secondary structure of proteins.
In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where two regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
So secondary structure is based upon hydrogen bonding. The hydrogen bond between parts of the amino acid sequence causes the molecule to begin to fold on itself in either the alpha helix, which is kind of a corkscrew or coiled shape arrangement, or into the beta pleated sheet, which is kind of a folded, corrugated looking appearance in the protein. So far we have the amino acid sequence, that's the primary structure, and now we have a secondary structure as a result of hydrogen bonding within the molecule, and the two ways that forms is through the alpha helix and the beta pleated sheet. Now let's look at tertiary structure.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
So tertiary structure is the result of ionic bonding, hydrophobic interactions, hydrophilic interactions, covalent bonding between sulfur atoms. All of those different types of interactions that we have discussed can all be found within one protein structure. Here's the tertiary structure of transthyretin. We see the ribbon as the primary structure in this example. Then we see the alpha helices and the beta pleated sheets. And then the remaining foldings that we see would be the tertiary structure. Now let's look at the last level of structure, which is the quaternary structure.
Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of 4 chains, make up each hemoglobin molecule.
So the final structure of our transthyretin protein are 4 identical polypeptide chains that are held together by similar chemical interactions that we saw in the tertiary structure. Things like hydrophobic, hydrophilic interactions, ionic bonding, covalent bonding at sulfur groups, and so forth. All of those would go into making the full protein with all 4 parts associated with it. So here we see them step by step again. Remember, primary structure is the amino acid sequence. Secondary structure is a result of hydrogen bonding, leading to either the alpha helix or the beta pleated sheet. Tertiary structure is a result of other kinds of chemical interactions that fold the protein even further. And then quaternary structure is the combination of many polypeptides coming together to form one single functional protein. So here's our transthyretin again, and we can see the 4 individual polypeptides that make up that protein. Sometimes a protein shape is going to be altered, and when it is, it's no longer functional. This is called denaturation, and this will cause a polypeptide chain to unravel and lose the shape and its function. Proteins can be denatured by changes in salt concentration, temperature, pH. These are all agents that will cause the interactions, the chemical interactions of hydrogen bonding, ionic bonding, hydrophilic, hydrophobic interactions to shift from the normal protein structure to a more unraveled, or non-functional protein. In some cases, we can renature that protein back to its original structure, but many times in a case like temperature, we can never return the protein back and it simply remains non-functional. So far, we know that the chain of a protein, the amino acid sequence, folds into a complex shape, and the shape depends on which amino acids form that sequence. The folded shape is held together by hydrogen bonds and other interactions associated with the R group, and then some, not all, but some proteins come together with more than one polypeptide chain to have quaternary structure. Okay, I'll see you soon. Ta-ta!